Hi everyone, welcome to today's live broadcast, PD-1 Cancer Immunotherapy, presented by Dr. Gordan J. Freeman. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Here's how the presentation works. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure to follow up with you by email. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right of the Q&A, on the top right, or in the Q&A button on the lower left. I would now like to introduce today's speaker, Dr. Gordan J. Freeman. Gordan J. Freeman, PhD, works in the Department of Medical Oncology at Dana-Farber Cancer Institute and is Professor of Medicine at Harvard Medical School. Dr. Freeman earned his BA in Biochemistry and Molecular Biology and PhD in Microbiology and Molecular Genetics from Harvard University. His research has identified the major pathways that control the immune response by inhibiting T-cell activation, PD-1-PDL1 and B7-2-CTLA-4, or stimulating T-cell activation, B7-2-CD28. In 2000, Dr. Freeman discovered PD-L1 and PD-L2 and showed they were ligands for PD-1, thus defining the PD-1 pathway and the drug target, block the interaction. He showed the function of PD-1 was to inhibit immune responses, and that blockade enhanced immune responses. He showed that PD-L1 is highly expressed on many solid tumors such as breast and lung, as well as some hematologic malignancies, and allows these tumors to inhibit immune attack. He received the 2014 William B. Cooley Award for Distinguished Research in Tumor Immunology for his work that led to development of PD-1 pathway blockade for cancer immunotherapy. I will now turn this presentation over to Dr. Freeman. Dr. Freeman?
Good morning. I'm Gordan Freeman from the Dana-Farber Cancer Institute, and I wanted to talk today about PD-1 immunotherapy. Could you put up the talk slides? I only see the disclosure slides right now. Good. Okay. Good. So these are my disclosures. So immunology has offered hope for curing cancer for the last 100 years, but you couldn't go into your doctor's office and get an effective immunotherapy. Now, that's changed in the last couple of years. And what's different now? And what's changed is we have a new strategy. And that new strategy is to block the pathways used by tumors to inhibit the anti-tumor immune response. And this has been termed checkpoint blockade by Jim Allison. Now, T cells are the white blood cells that can kill cancer cells. And when a disease starts, you only have a tiny number of T cells that can attack that disease. So for instance, right now, you probably only have 1,000 T cells that could attack Zika virus. And that isn't an effective fighting force. But once the immune system recognizes a disease, the T cells become activated and start to frantically proliferate. And over a week, they go from, say, 1,000 T cells to millions of T cells. And millions of T cells can orchestrate and mount an effective immune response. Now, T cell activation requires two signals. The first signal is provided by the T cell receptor interacting with peptide antigen on the antigen-presenting cell. And that gives the response specificity. But T cells need a second signal, a co-stimulatory signal for full activation. And that's what I studied. And the surprise over the last 20 years is that there are both positive and negative second signals. Now, I got into this field through studying the B7-1 and B7-2 molecules, which are the receptors for CTLA-4 and CD28. Now, when the human genome began to be sequenced, we looked for what molecules were cousins to the B7 molecules, and we identified two that we called PD-L1 and PD-L2. And with Clive Wood, we showed these are the binding partners, the ligands, for a molecule called PD-1. And when PD-L1 or PD-L2 bound to PD-1, they resulted in tyrosine phosphorylation of the cytoplasmic domain of PD-1, and this recruited phosphatases such as SHP2. And this led to reduced activity of the proximal signaling kinases of the T cell receptor and CD28 pathway. And this results in reduced TCR signaling, less cytokine production, reduced target cell lysis. It also has effects on altering lymphocyte motility and metabolically reprogramming the T cell. Now, this work was published in 2000, and it identified the drug target, identified drugs that would block the interaction of PD-1 with PD-L1. Now, PD-1 was cloned in the 1990s by the Hanjo lab from a CD3 activated T cell hybridoma undergoing activation-induced cell death. And so they called it program death 1. But this is somewhat of a misnomer because it doesn't directly activate caspases and cause cell death or apoptosis like the CD95 or FAST molecules. It can have an indirect effect on cell death by reduced cytokines, less survival factors like BCL-XL, or more BIM, as shown by Dong. Now, we have negative signals like PD-1 for an important reason. And Arlene Sharp and Tasuko Hanjo's lab have studied the knockout mice. And they show that the PD-1 pathway is important in maintaining tolerance. It tunes down the immune response after the elimination of disease, and it prevents too strong an immune response from damaging tissues. Now, soon after we cloned PD-L1, we identified PD-L2 as a second ligand. Now, we knew we had a pathway that inhibited immune responses, but we discovered something surprising. We found that PD-L1 was expressed on solid tumor cell lines, not unlike a typical immune molecule. And this slide shows PD-L1 expression on breast cancer cell lines. And we said that this might represent a shield against the immune system by cancer. And this has proven to be a remarkably accurate prediction. When people began to look at PD-L1 expression on actual tumors, first shown by Lai-Ping Chen, we found that PD-L1 was expressed on about 30% of solid tumors and selected hematologic malignancies. And this inhibited the anti-tumor immune response. So here in brown is the PD-L1 expression, And you can see expression on the surface of kidney tumors, or on the right, expression on a lung cancer. So PD-L1 turns off the immune response. But in a cancer patient, the whole immune response isn't turned off. A cancer patient can still mount a fine immune response against the common cold. For instance, there's no PD-L1 expression in the nasal cavity, and so there's a good immune response. However, locally, in the cancer in the lung, you can see there's a lot of PD-L1 expression, and this turns off the local immune response. So immunologists have tried to stimulate an anti-tumor immune response for many years, but it hasn't worked. And one of the reasons that directly stimulating the immune response hasn't worked in real patients is that the immune response has gotten ahead. And if a T cell recognizes a tumor, it becomes a little activated, but it makes some gamma interferon. And that is seen by interferon receptors and upregulates PD-L1 expression. So once the tumor gets ahead, the very act of recognizing the tumor turns off the immune response. So PD-L1 expression can come in several different patterns. And this is a pattern which has been termed adaptive immune response by Janet Taub. And here what you see in a melanoma, in brown on the right, is PD-L1 expression on the periphery of the melanoma, but not in the middle. And if you look on the left, you see in brown PD-1 expression on T cells. And these T cells are held up at the border of the tumor and don't really enter it very much. So this locally turns off the T cells. Now, we had identified the drug target, so we made antibodies against PD-L1 and PD-L1, as did multiple other groups. And we showed that if you block the interaction of PD-L1 with PD-1, that you activated the immune response. You got an enhanced immune response with increased T cell production of cytokines like gamma interferon and increased killing of tumor cell targets. So where does immune checkpoint blockade function? Jim Allison pioneered CTLA-4 blockade, and this is blockade of an immunoinhibitory pathway, the interaction of B7 with CTLA-4, which primarily occurs in the lymph node where there's significant B7 expression on antigen-presenting cells. In contrast, we had a different idea. We wanted to block the immunoinhibition by the tumor in the tumor microenvironment. So we wanted to block PD-L1 expression on the tumor, on infiltrating myeloid-derived suppressor cells, stromal cells, antigen-presenting cells. And so PD-1 blockade works within the tumor and the tumor microenvironment. Now, multiple pharmaceutical companies have developed agents that block the PD-1, PD-L1 interaction, and these include nivolumab by Bristol Myers Squibb and pembrolizumab by Merck, or atezolizumab by Roche. In addition, multiple other companies have made drugs that block the pathway, and these have been enrolled in clinical trials. Now, I'm a PhD immunologist, not a physician, and this slide was of a kidney cancer trial was kindly provided by Chuck Drake. And the what this shows is the analysis of 34 cancer patients with the PD-1 antibody nivolumab. Now, what we're looking at here is each line follows the growth or diminution of one tumor, and it follows the change in tumor size. Now, some tumors continue to grow in the presence of PD-L1 and go on to a different therapy. In contrast, some of the lines go down, the tumor is reducing in size, and it's a good response. If the tumor shrinks in size by 30% or more, this is called an objective response. And in this clinical trial, 29% of the patients had an objective clinical response. What was impressive is that in this trial, all the patients stopped therapy after about 2 years. And after stopping therapy, the tumor didn't immediately regrow. You can see in the red circle that the tumor stays small, and there's survival even off drug. Now this slide shows the response rates to PD-1 and PD-1 inhibitors in multiple clinical trials. In fact, over 250 clinical trials up to 2015. Now, the first study was published by Julie Brommer and Suzanne Topolian, and this was in multiple tumor types and showed positive responses. You see about a 35% response rate in melanoma, 30% in bladder, 20% in a number of tumor types. PD-1 therapy doesn't work in all tumor types. For instance, there's a pretty low response in colon. There's very good response in Hodgkin's. So in green now are the tumors where the trial results have been successful enough to lead to FDA approval of the PD-1 drug for that tumor type. So there's approvals now in advanced melanoma, bladder, lung, head and neck, kidney, and Hodgkin's lymphoma. Just to make sure that I'm on, would the moderator just please give me a typed message that everything is good? All right, good. Good to hear it. Okay, so I'd like to emphasize that PD-1 therapy is different from chemotherapy. It's not a cell poison. You're not trying to directly kill the tumor cell. Instead, you're trying to let the immune system become activated and the immune system kill the tumor. So the PD-1 therapy is well tolerated. There's no blood cell count decline, no hair loss. There is some nausea. It's got an acceptably good safety profile. The most serious adverse events are autoimmune mediated, like pneumonitis or colitis, immune attack against some organ in the body. This occurs in about 10% of patients. And so this is different from the complications of chemotherapy. So physicians are really going to have to learn to manage a different spectrum of adverse events than those seen in chemotherapy. Now, the PD-1 antibody is delivered with about a half-hour intravenous drug infusion in an outpatient basis. So this is potentially, if physicians become comfortable with it, community hospital medicine. Now, the other thing that's notable about the PD-1 therapy can be, if it works, the quality of life can be good. This is some studies from Barmel and Pickard presented at SITC. And what you see is this looks at the quality of life of a lung cancer patient who's doing well on chemotherapy, docetaxel. And the quality of life of a chemo patient is basically the quality of life of a lung cancer patient. In contrast, if you respond to PD-1 and are doing well, your quality of life returns pretty much to the population norm. And one of the authors was quoted as saying, this suggests that prolonged survival occurs with a resumption of normal life, which is a real improvement. Now, another notable thing is that checkpoint blockade has been studied both in elderly and younger patients, the cutoff being about 65 to 70 years. And about 3,000 patients were analyzed. And the hazard ratio, the odds of success and complications, are about the same in the younger patients or the older patients. So it can work equally well in the aged. And this can lead to successes such as a 90-year-old with metastatic melanoma and 4 brain metastases who was treated with the PD-1 antibody pembrolizumab. Now, predictive biomarkers are really essential to getting the right treatment to the right patient. And the first groundbreaking study by Topolian was black and white. On the left here, it showed that if the tumor was PD-L1 positive, there was about a 36% response rate. In contrast, if the tumor was PD-L1 negative, there were no responders. Multiple other studies have shown that it's not quite so black and white, though the general trend is that way. So if the tumor and the tumor microenvironment expresses PD-L1, there's a higher response rate, but there's some modest level of response in patients whose tumors are PD-L1. So PD-L1 expression isn't a good enough biomarker to say yes, no. We need something better, though it can guide therapy. So it used to be that PD-L1 immunohistochemistry was difficult and artsy. However, a lot of effort has been put into developing good antibodies, and there are really now 5 good, widely available immunohistochemistry antibodies for PD-L1. The original groundbreaking work is done with 5H1 from Chen, but there are now antibodies available from Merck, BMS, Roche, and in addition, antibodies from my lab and cell signaling technology, all of which work well. Some of these see the outside of the PD-L1 molecule, whereas some see the inside. So for instance, the 9A11 and the 142 antibodies see the inside cytoplasmic domain of PD-L1 and can give good immunohistochemistry, as shown in these slides, where you can see nice PD-L1 expression on the cell surface. Now, with Sabina Signoretti, we looked at PD-L1 immunohistochemistry and tried to see whether there was concordancy or discordancy between primary tumor and metastasis in renal cell carcinoma. And we looked at 53 cases, and what we see is that 33 of those cases are negative in both the primary and metastasis, and so are concordant. Now, of the 20 positive cases, what we saw is that 9 had a PD-L1 expression, both in primary and metastasis, and so were concordant, but that 3 had expression only in metastasis, and 8 had expression only in the primary. So what Sabina emphasizes is that PD-L1 positivity was heterogeneous and almost exclusively detected in the high nuclear grade areas of the tumor. An assessment as a predictive biomarker may require analysis of metastatic lesions, and pathologists should really select the high-grade tumor areas to avoid false negatives. Now, in addition, PD-L1 expression can be heterogeneous within the same tumor. Here you see little PD-L1 expression in a low-grade area of a kidney cancer, but a lot of expression in a high-grade area. Now, when the immune system attacks a tumor, what does it see in the tumor to attack? And what the tumor sees is changes in the tumor cell. And these are primarily protein coding changes brought about by mutations which change the amino acid in the protein. And these are called tumor neoantigens. And there are really 2 evolutionary processes going on in cancer. There's rare mutations, such as in, say, BRAF or RAS, which are driver mutations which drive the proliferation of the tumor. But there are many different passenger mutations which can change protein coding sequence and lead to tumor neoantigens, as indicated by these symbols in the tumor. But there's also another evolutionary process going on in the tumor. And this has really been pioneered by Bob Schreiber. And what he's shown us is that the immune system eliminates many early tumors, but the tumors that grow and become a medical problem have learned how to evade the immune system by expression of molecules like PD-L1 or IDO, TGF-beta, IL-10, loss of MHC class 1 or 2, so that there's really two evolutionary processes going on. And one thing that's really notable is that if you target just a driver mutation, if you target just one thing in a tumor, the tremendous mutational heterogeneity of the tumor will learn how to evade it and become resistant. What's good about immunotherapy is it engages the immune system, which has billions of T cell receptors, billions of antibody molecules, and thousands of pattern recognition molecules, which can recognize the changes in the tumor. So the immune system is also an evolving system, which can keep up with the evolution of a tumor. Now, on the right here is a patient's response in the melanoma to the BRAF targeted kinase inhibitor, vemurafenib. The wonderful thing about the TKI is if you have the mutation, you're almost 100% likely to respond to it. But this response is evaded because it's just one target. And after a year or so, the tumor becomes resistant to that one targeted therapy. Some of the newer TKIs can be better and give 3 or more years of benefit. In contrast, on the left is why there's so much enthusiasm for immunotherapy. The green line analyzes about 2,000 patients who've been treated with the CTLA-4 antibody ipilimumab. And what you see is that patients are only treated for about 1 year with the CTLA-4 antibody and then stop treatment. About 20% of patients by year 3 are alive. And if you follow these patients out for years 4, 5, 6, 7, 8, 9, out to 10 years, they're still doing well, they're still alive. And this is what let's talk about a cure. Now, PD-1 hasn't been analyzed for as long. In the blue line is a trial reported by Steve Hody and Jed Walchuk, and it shows that PD-1 also has a survival benefit of about 35%, and that this is enduring, and it follows patients out up to about 5 years. Now, CTLA-4 has only been clinically approved as a single agent in melanoma. In contrast, PD-1 has now been approved in 6 tumor types with successful clinical trials in multiple other cancer types. So PD-1 is a broader immunotherapy. Now, understanding in immunology and genetics has helped scientists identify groups that will respond well to the PD-1, PD-L1 therapy. Margaret Shipp at the Dana-Farber Cancer Institute has analyzed Hodgkin's lymphoma, and what she's shown is that almost all Hodgkin's lymphoma genetically amplify the PD-L1 and PD-L2 chromosomal locus. So all Hodgkin's lymphomas express a lot of PD-L1 and PD-L2, and they really depend on it. And if you block PD-1, you get an 87% response rate in Hodgkin's lymphoma. The group at Johns Hopkins reasoned that since neoantigens are so important, that if you had a tumor type which had a defect in DNA repair, which would lead to lots of DNA damage and lots of mutations, such as the microsatellite stable, unstable type of colorectal cancer, that you'd have a highly mutated tumor with a lot of targets. And indeed, their prediction is spot on. and you get a 62% response rate in tumors with defects in DNA repair. Now, it's been estimated that about 25% of human cancers may be caused by viruses, such as papillomavirus or Merkel cell polyomavirus. And these express foreign viral antigens. And the immune system is really good at seeing foreign. And so there's good response rates in Merkel cell or head and neck tumor. And really, we want to creatively think what other tumor cell types may have a defect that makes them highly responsive to immunotherapy. Now, the real challenge now is to only 60 to 80% of patients don't get benefit from PD-1 inhibitors. And the goal now is to bring clinical benefit by immunotherapy to these other patients. And this is going to come about through combination therapies. Now, why did the T cells need PD-1 to blockade to attack the tumor? And the reason is that the anti-tumor immune response is a years-long struggle. The immune system has tried to attack the tumor all these years, but the tumor has evaded the immune attack, and the T cells have tried to attack, failed, and become exhausted. And this idea comes about through a collaboration with Rafi Ahmed and John Wary. And T cell exhaustion, however, is more than just expression of PD-1. Exhausted tumor infiltrating lymphocytes can express multiple immunoinhibitory receptors, including all the ones shown on this slide, including CTLA-4, LAG-3, TIM-3, TIGIT. And these are all druggable targets that scientists and pharma companies are developing drugs for tumor immunotherapy. Now, so the future is clearly combination therapy, building on a foundation of PD-1, but adding other checkpoints such as CTLA-4, LAG-3, or TIM-3, or activating molecules like CD137, OX40, or ICOS, or stimulatory cytokines such as IL-2 or IL-12. Now, Alan Corman's group first shown the effects of PD-1 with CTLA-4. And I'm going to show you some combination studies in a mouse model of glioblastoma, because this is a tough model in the brain with immune privilege where the immune system doesn't get too easily. So this work was done with David Reardon and Praful Gokhale, Sarah Klein, and the tumor is implanted into the brain of a mouse, and it develops. And after about 2 weeks, we begin therapy with CTLA-4 and or PD-1 antibodies. And what we see is that in this late model, that CTLA-4 alone gives some benefit, but not long-term survivors. PD-1 antibody gives about 50% survivors, but the combination of PD-1 and CTLA-4 gave 7 out of 8 survivors in this experiment. Now, this is basically visualizing the tumor in the brain of a mouse with luminescence. And what you see is the tumor in the control group begins to develop and then grows very large. In contrast, in the treatment group with PD-1 and CTLA-4, the tumor begins, it grows larger, but then the immunotherapy kicks in and eliminates the tumor. The checkpoint blockade causes much more immune infiltration of T cells into the tumor, as indicated by these brown T cells in the brain tumor. And in addition, the checkpoint blockade has an effect on T cells. The PD-1 results in a lot more NK cells in the tumor. The CTLA-4, not so much. So we also saw that in the brain, the NK cells express more CD86 molecules as a reflection of activation. So we wondered whether NK cells were critical in the brain. And we used antibodies to eliminate CD8 cells or NK cells in the brain. Now the blue here shows the effect of PD-1 antibody alone. And if you add NK 1.1 in the green with PD-1 to eliminate the NK cells, you show the treatment is just as effective. So NK cells aren't absolutely essential in the brain, though they may contribute. In contrast, in this purplish, you see that if you eliminate CD8 T cells, that the treatment doesn't work. So CD8 T cells are essential. And in other experiments, Sarah also showed that CD4 cells are essential for the success of immunotherapy. So it requires all the arms of an immune response. So though we think NK cells are important, one of the obvious evasion mechanisms to immunotherapy might be loss of MHC by the tumor. But the immune system has lots of ways to attack. And if a tumor loses MHC, it becomes much more susceptible to NK cell lysis. And here, with Roberto Bellucci and Jerry Ritz, we've shown that NK cells do express PD-1, and that if you block PD-1 on NK cells, you get better lysis by the NK cells. So the PD-1 blockade can enhance the immune response by multiple arms of the immune system. So we wondered whether we could use the wonderful TCGA database RNA expression of tumors to help us guide immunotherapy development. And there are a number of other genes in the B7 gene family, in addition to PDL1 and PDL2. These include B7H3 and H4, about 20 members of the butyrophilin gene family, VISTA, icosligand, HHLA2, and others. And so we asked, are these expressed by tumors? And I got Sam Freeman to analyze expression of these molecules in normal kidney, shown in blue, versus kidney cancer in this reddish color. And what he found was that kidney cancer was rather extraordinary. Over on the right, what you can see is there's much more CD8 expression and CD4 teen expression, reflective of a lot of infiltrating immune CD8 killer cells and myeloid cells. And when you look at the B7 family, what you see is that there's more PDL2, more B7H3, surprisingly less B7H4, more VISTA, more HHLA2. And so these are all now targets for immunotherapy of kidney cancer. And we went on to do some immunohistochemistry for the VISTA molecule, and we see that on the left, VISTA can be expressed by the kidney cancer cells. On the right, it can be expressed just on the infiltrating immune cells and not on the kidney. So you really need to look at the actual kidney to distinguish where the molecule's expressed. Now, there are many more combinations possible, including oncolytic viruses, chemotherapy, radiation therapy, targeted therapies. Epigenetic modifiers, such as HDAC inhibitors, are very promising. Blockade of phosphatidylserine. In addition, once you block immune suppression by the tumor, this really enables other therapies, such as vaccines or stimulations by STING or TLR agonists, GM-CSF or interferons, or CD40 agonists. The real key is to block the tumor from turning off the immune system. Other combinations include depleting other immunosuppressive cells in the tumor microenvironment, like MDSCs or Tregs, or blocking other immunosuppressive molecules like IDO or TGF-beta. In addition, there's a lot of creativity going into adding back immune cells, such as CAR T cells or TCR-engineered T cells or NK cells. And these T cells, such as CAR T, are T cells, and they can, over time, become exhausted. And with Wayne Marasco's group, we've shown that if you include PD-L1 antibody with a CAR T cell, that this lets the CAR T cell work longer and better. In addition, a number of classical therapies, such as surgery or radiation, can work together with PD-1 blockade. In particular, many types of chemotherapy might be thought as just killing all dividing cells. But some have actually been shown to work with immunotherapy. And this shows basically a paper we've done with Christina Viersky and Mikhail Pitet. And this looks at a genetic model of lung cancer. And here in the dark blue is growing tumor in the lung. So on the upper left is large tumor growing in the untreated mouse. Now this tumor didn't respond to PD-1 and CTLA-4 therapy, and it responded weakly to the combination of oxaliplatin and cyclophosphamide. However, what was surprising, they discovered, was that when you killed the tumors by the oxaliplatin-cyclophosphamide combination, that caused the tumor to die in a way which was stimulatory to the immune system. Antigen-presenting cells took up the tumor, became activated, and presented it to the immune system. And when you blocked PD-1 and CTLA-4, that combination then really succeeded and did well. So there's chemotherapy combinations that can combine with immunotherapy. So what happens when tumors respond to PD-1 immunotherapy, but then develop resistance? And this work comes from the TIM-3 molecule that we discovered with Jason Galea, Vijay Kuchar, and Rosemary DePrive. And we've shown that TIM-3 has two primary ligands. Phosphatidylserine binds to a deep binding pocket, and Vijay Kuchar's group has shown that galectin sugar binding molecule binds to sugars on the glycosylations of the TIM-3 molecule. And we think these work together to transduce TIM-3 signals and inhibit immune responses. And with Shohei Koyama and Esra Akbe, Kwok Wang, and Peter Hammerman, we showed that in patients where therapeutic PD-1 blockade loses efficacy, that it's associated with upregulation of additional immune checkpoints. And this work was again done in a genetic model of lung cancer. And Shohei and Ezra treated this genetic lung cancer model with PD-1 antibody, and the tumor was responsive for a number of weeks, but then began to become resistant and grow again. And they looked at RNA expression in the untreated versus the resistant tumor. And what they found is the resistant tumor developed a lot of expression of the TIM-3 molecule, or LAG3. And so these are additional targets. So they went on and showed that in the lung cancer in the mouse, that in the untreated mouse, in the clear circles, there was little TIM-3 expression. In the sensitive tumor, there was just a little TIM-3 expression. But as the tumor became resistant to PD-1, it developed a lot of TIM-3 expression. Now, Peter identified some lung cancer patients who had initially responded to PD-1 therapy, but then become unresponsive and developed resistance. Now, the primary tumor in blue didn't express much TIM-3. And in clear, the control lung infusion didn't affect moderate amounts of TIM-3. But when the tumor became resistant in red, it expressed a lot of TIM-3. So Ezra and Shohei went back to the mouse model, and what they showed is that if a mouse lung tumor became resistant to PD-1 therapy, but that you then added TIM-3 therapy in the red, that you'd get additional clinical benefit, and the mouse would do well for a while before declining. So the combination of PD-1 and TIM-3 can benefit when you develop PD-1 resistance. So in beginning to wrap up, I'd say it's really an exciting and great time to be an oncologist because it's a better time for patients. PD-1 and PD-L1 works in a wide range of tumors with a moderate percentage of responders and good safety profile. So it's a good foundation to build on. And it's attracted now a multitude of researchers in academia and pharma. And we know we can do better, and multiple preclinical and clinical studies are now identifying ways to do better. Really, human creativity has been unleashed, and we're really learning to do better. We need to identify who will respond to PD-1 blockade, and what are the mechanisms of failure to respond, because it doesn't work in everyone. It works in about 20%, which means it doesn't work in about 80%. So does it not work because there are other immunoinhibitors, or failure of immune cells to infiltrate the tumor, or that the tumor doesn't have enough mutations to generate good neoantigens? Also, what are the secondary mechanisms of failure to respond? Expression of other immunoinhibitory receptors like TIM3 or loss of MHC molecules? So I think the future of cancer therapy decisions is first going to be guided by precision medicine. We're going to sequence tumor genomes. And the sequence of tumor genomes of an individual patient, first they can tell us those populations who could respond really well to PD-1 therapy, which patients have PD-L1 and L2 genomic amplifications, which have defects in DNA repair, and so lots of mutations, such as the MSI colorectal or other MSI tumors, which have viral genomes and have good foreign antigen targets. All those patients will respond well to PD-1 therapies. In addition, the sequences will identify which oncogenes are drug targets, and such as for BRAF targeted kinase inhibitors. And it'll also identify neoantigens in the tumor, which could be made into vaccines to stimulate the immune response. Now, the pathologists are going to look at the tumor and come up with a tumor immune evasion score. They're going to tell us how much PD-L1, PD-L2, IDO, and other immunoinhibitory molecules are expressed by the tumor. And that'll let us choose the best immunotherapy and combine it with the best targeted therapy or vaccine. So I think this is the future of cancer therapy. Now, these are the people in my lab who've done the work. Not pictured here are the pioneers in the lab, Julia Brown and Kwafeng Kai. Yanping Xiao and Kathleen Mahoney, Sanhong Yu are pictured here, and did the glioblastoma and immunohistochemistry studies. Apurvi Chaudhry has studied the interaction of PD-L1. Sarah Klein, the glioblastoma model. Sha Bu has studied TIM3. Ping Hua, Bao Guangzhu, Yahui Huao have all helped develop the PD-1 pathway and other immunoinhibitory pathway antibodies. Lily Kai is a great undergraduate who studied other combination therapies. This work has been a wonderful collaboration from its earliest days, really beginning with Clive Wood and Tsuko Hanjo in the identification of the PD-L1, PD-1 interactions, showing the pathway was inhibitory, and identifying the drug target. It's been a wonderful collaboration with Arlene Sharp, identifying via the knockouts and in vivo studies the functions of the pathway, with Rafi Ahmed and John Wary on exhausted T cells, with Vijay Kuchar on the TIM3 molecule, with Vicky Busiadis on the biology from the earliest days, with kidney cancer with David McDermott and Michael Atkins, ovarian cancer with Kumar Duraswamy, George Kukos, and at the Dana-Farber, Hodgkin's lymphoma with Margaret Schiff, tumor immunotherapy with Glenn Dranoff, tumor immunotherapy with David Reardon, Kwok Wong, CAR T-cell with Wayne Marasco, and mechanisms of immune evasion with Peter Hammerman. So I think even better days are before us. Thanks, and I look forward to your questions.
Thank you, Dr. Freeman, for that informative presentation. Okay, now we want to get right to your questions and input. So here's a reminder how to let us know what you have to say. Questions, etc., can be submitted via the Q&A button at the lower left. We will try to get to all of you, but if not, we will be sure to follow up after the broadcast. Our first question is, Dr. Freeman, are there any predictive laboratory biomarkers of autoimmune adverse effects anti-PD-1 therapy?
Oh, there are no markers for which would indicate development of autoimmunity. In initial clinical trials, patients with autoimmunity such as multiple sclerosis aren't included in immunotherapy trials. Some have been included, now that the drugs are approved in some tumor types, some patients have been included. There's just a very limited clinical experience. One didn't get any exacerbation of autoimmunity. I think one did. Exacerbations can be treated with anti-inflammatory agents like corticosteroids and others. It's really an active area that needs investigation and safety assessment.
Question number 2. Do PD-1 inhibitors impact the propagation potential of human tumors in xenografts in NOG-SCID or NOD-SCID studies in mice?
In general, an immunotherapy depends on an intact immune system. And the human tumors, when they're put in a mouse, have to be put in a mouse which has no immune system. So it's you can't study immune response in that model. If you also add human immune cells, you can make some attempts to study the anti-tumor immune response. It's an area of very active research where the leaders in the field are developing better models, and really there's additional work needed there to make it better.
Next question. For pathologists, should all laboratories use PD-1 IHC staining in cancer specimens? Is that financially feasible?
I don't think the immunohistochemistry is a big part of the cost. We need better biomarkers because the PD-L1 and immunohistochemistry is only suggested. There's been a lot of press now on the use of the PD-L1 immunohistochemistry in lung cancer. In lung cancer, in a trial that was done where the cutoff was 50% PD-L1 expression on the tumor, PD-1 antibody had a measurable success if the tumors had 50% more or more expression of PD-L1. In contrast, if the cutoff was lower, if the tumors only 5% expressed PD-L1, the clinical trial wasn't successful. So PD-L1 expression may be used to guide who is most likely to respond. Nevertheless, there's a low but real expression or success rate in tumors that don't express PD-1. It's an area that, again, is under intense investigation. Additional biomarkers are needed, like tumor neoantigen load, the extent of CD8 infiltration into the tumor, or we need other additional combinations of biomarkers which predict treatment success.
Next question, Dr. Freeman. Is immunotherapy first, middle, last, or combinatorial cancer management?
It depends on the tumor type. Melanoma is the most responsive cancer type to immunotherapy, and actually PD-1 is already first-line therapy there. In other tumor types, it needs to be tested to see whether it's a good, successful first-line therapy. Chemotherapy can have a high percentage of response rate, but it totally succeeds for some patients, but it fails and the tumor develops in others. So we need basically trials that compare immunotherapy to the best successful standard therapies of chemotherapy or whatever the best therapy is, to compare and see whether the immunotherapy is right to start with or whether other therapies are right to start with.
Dr. Freeman, do PD-1 inhibitors, MABs, work better by injecting into the solid tumor as compared to given systematically?
No, the drug delivery is always within the drug delivery is always with an intravenous injection. There have been I think I don't think patients like the idea of multiple needle sticks into a tumor, and the intravenous infusion spreads it throughout the body and can target metastases at different sites also. I know there's a lot of creativity going in to the idea of making antibody agents which only open up and reveal themselves when they're actually in the tumor.
Next question. Do PD-1 inhibitors impact the propagation potential of human tumors? Oh, we already asked that one. I apologize. Let's do question number 6. For a patient who is seeking anti-PD-1 immunotherapy, which steps should he take? What if his oncologist does not know about immunotherapy? What should he do?
I probably go to one of the more research-oriented medical centers, of which there are multiple ones around the country. Those have physicians who are now have some experience with immunotherapy and managing patients. There's also a very good site called clinicaltrials.gov, which you can find on the web. If you find that site, basically all the clinical trials in the United States are listed on that site. And you can search it. You can put in, say you had pancreatic cancer. You could basically put in pancreatic cancer in the website, and then you could put in different agents. You could put in PD-1, TIM3, LAG3, vaccine, and the website will then search all the ongoing clinical trials and come back with a list of where the clinical trials are being conducted and who to contact to get on that clinical trial. So clinicaltrials.gov is a wonderful site for identifying trials which are investigating your tumor type.
Thank you so much, Dr. Freeman. I'm sorry, we don't have time for any more questions. We've had great questions coming in. I would like to thank Dr. Freeman one more time. Dr. Freeman, do you have any final comments for us?
It's exciting times. I think that we're having success, and we know how to have even greater success.
Thank you, Dr. Freeman. We remind everyone that today's webcast will be available for on-demand viewing on our website through February 2017. We will let you know when it's been posted, and we hope you pass that information on to any colleagues who couldn't take part in today's broadcast. That's all for now. We appreciate you joining us. We hope to see you again soon. Bye.